A great lesson in store for you, talking about Cytopoint and specifically what is IL-31. I read a lot to prepare for this lesson, and a lot of those readings are included in your downloads, so make sure you're checking that out because honestly, it was a lot of fun to read about IL-31 and the role that it plays in allergic skin and ear disease in dogs and the immune system and just what it is. It's a cool cytokine. So I really hope that you enjoy the worksheets and the extra credit readings and this lesson. So let's get started. Cytopoint, we already know, is a canonized monoclonal antibody against IL-31, and it was approved by the USDA in 2016 for canine allergic and atopic dermatitis. And we know that Cytopoint is an antibody. Antibodies are proteins that are created by the immune system to target and neutralize antigens. So if Cytopoint's an antibody, well, IL-31 is its antigen. What's IL-31? Well, we know that it's a cytokine, and cytokines are small proteins or soluble mediators that are helping with cell signaling and communication. And this is how I boil it down. Cells produce cytokines and cytokines produce effects. It's like not that deep. They're just messengers that are helping cells communicate. And so here's our first question. What cells produce IL-31? If cells produce cytokines and cytokines produce effects, well, our first step in understanding IL-31 is understanding what kind of cells are making it. So there's basically four, but mainly one that we need to think about. The main producer of IL-31, these are activated T cells. Now, we've already learned about the Th2 shift. These are cells that are part of the T cell domain, and they're leaning toward a certain profile where they're creating more cytokines to fulfill the allergic criteria, or to say, oh, we're generating an allergic response here. We need to create messages that are in line with that allergic response. And one of the messages that these activated T cells are going to produce is going to be the cytokine IL-31. And so this cytokine is produced largely by activated T cells that have been stimulated and are in the kind of rolling process of the allergic cascade. Okay, so it should be no surprise that then mast cells and eosinophils, two other big players in the allergic response, are also capable of producing IL-31. And then finally, dendritic cells. Well, these are antigen-presenting cells. No surprise there. If they're finding antigens like ragweed pollen, they should be able to create some of this allergic cascade as well. So all of these cells are directly in charge of creating that Th2 shift and that allergic cascade, which we see manifest the phenotype of allergic skin and ear disease. And so we have IL-31, IL-4, IL-13, and many other cytokines that are part of that cytokine milieu called the Th2 shift. Now we remind ourselves what exactly that is. This is taking a naive T cell and creating a switchover that forces the response because of the environment locally and the types of cytokines present and the body's predisposition and genetics and all of the factors that go into pushing a group of cells in one direction over toward a shift to continuing in that direction, and that is the Th2 shift. And we see that more with allergic disease. And when you think about the cytokines that are involved in allergic disease, there are a lot of them, and IL-31 is one. So when we're thinking of IL-31, I love it, and I want to say, oh my God, what an amazing cytokine, but it doesn't need to be overexploited. We don't want to put it on, again, we'll talk about pedestals when we talk about itch, but it's only one part of a very big problem. Sometimes I describe IL-31 as an annoying office lady. So we think about atopica being something that really can target inflammation. So I think of that as a vice president. I think of Apoquel like a middle manager because it's really in that company of allergy, not the broader company of inflammation. And then I think of IL-31 or Cytopoint as targeting an annoying office lady. This is one of many cytokines that are involved in the allergic cascade. And it's, yes, important, but I just want to make sure that we're all on the same page, that it's not the only cytokine involved in allergy. So cells produce cytokines. Activated T cells, allergic type cells are going to produce IL-31. Well, what's the effect of IL-31? Once it's produced, what does it do? Three main things. Itch being the most important. So IL-31 is a peritogenic cytokine. This means that when it targets its receptors, it's going to generate an itch response. When we think of pruritus, that's just another word for itch. Pruritus is a sensation in the skin. Isn't that interesting? Is it a skin sensation or is it a brain sensation? As a philosophy major, that's something I could have talked about for a very long time when I was like a junior in college. God, those were the days. Anyway, this leads to scratching behavior. You're going to have this generation of an itch message to the brain, and the brain is going to say, okay, body, it's time to itch. And when IL-31 is generated and it binds to its receptor, certain pathways like the JAK-STAT pathway are going to be activated. And when those receptors on these unmyelinated C fibers, nociceptive fibers are bound by IL-31, that itch message is going to be generated. So you can see here, when you have IL-31, again, it's going to target your neurons, and then again, that message is going to be sent to the brain. So the question is, okay, so this is like humans, we know this, IL-31. Dogs, IL-31, yes, the answer is yes. So we've looked at IL-31. Does it induce pruritus in dogs? Absolutely it does. So if you gave IL-31 to your pet dog, your dog would probably become itchier. And the reason is because IL-31 is going to bind IL-31 receptors, which are located on neurons, which generate itch responses. Just that simple. And then IL-31 also we know is going to be detected in the majority of dogs with allergic disease. So we do know that this is an important cytokine, again, in that profile for dogs with allergic skin and ear disease. So we know that IL-31 is a peritogen. Do you remember any other things that we talked about that were peritogens? Oh yeah, histamine. This is a table that has lots of peritogens that are listed here. So again, before we put IL-31 as being the only reason why itch is ever generated ever, we want to remind ourselves that there are lots of things that can generate an itch response, including histamine through the H1 and H4 receptor. So yes, IL-31 deserves a role when we talk about itch, but it is not the only peritogen ever. And so when we think about, gee, why didn't this work? Well, it just might not be that important for some dogs that are itchy. It might not be the main driver of their pruritus. In most cases, it is. It is an incredibly important part of the itch process and the itch cycle for dogs. That's why we have a therapeutic for it. But again, just put everything in perspective and remember it's one cytokine of many. All right, let's talk about the skin barrier. How does IL-31 and the skin barrier, do they have anything to do with each other? Well, yeah, they actually do. The presence of IL-31 in the skin barrier is really interesting to me and something that isn't talked about a lot when you kind of tune in. And I honestly, before having this lesson, didn't really ever hear anyone mention the role of IL-31 in the skin barrier. And to me, the skin barrier is one of the most interesting things that we can target when we're treating our allergic patients. So the skin barrier is composed of cells and cell junctions and proteins and lipids. And the role of the skin barrier is, there are many roles there, but it is largely to provide a barrier and to help keep water in and antigens out and, you know, all of the homeostatic mechanisms that the skin has, which is not what we're talking about here. What we're talking about is that the skin is a brick wall There's proteins and lipids that form the mortar, and then there are cells that form the bricks. And when we think about the important proteins that are in that mortar, well, filaggrin is one of the most important proteins. And wouldn't you know, IL-31 is actually going to impair or downregulate the production of filaggrin. And as a result, you have an impacted skin barrier homeostatic ability because one of the main players of the mortar is downregulated or reduced. And this isn't just IL-31. This is all Th2 shift cytokines. So this is one of the reasons why, if you remember all the way back to our introductory lesson, we talk about the fact that dogs have two problems, inflammation and a skin barrier issue. Well, the skin barrier issue happens because of inflammation. But if you're helping to target inflammation and skin barrier, well, there you go. Doing them is really hand in hand. It's going to help target the problems that these dogs have. And so filaggrin is absolutely going to hurt the skin barrier. It's going to impair the skin barrier such that it now has increased permeability. So IL-31 is influencing skin barrier homeostasis by down-regulating filaggrin. Fascinating. It's also pro-inflammatory. So what the heck does that mean? Well, we see that IL-31, I called it an annoying office lady earlier, and that's absolutely true. The presence of IL-31 is going to create or call other chemokines, cytokines, other mediators that are involved in the allergic cascade and response to the scene. Now, if you remove IL-31, are you going to eliminate IL-4 and IL-13 as well? No. So it's just one small part of a very large cascade. But is IL-31 part of that cascade? Absolutely. It is a pro-inflammatory cytokine. So there are direct and indirect effects of IL-31. Inflammation, skin barrier changes, and itch. Itch being the most important. When we look at dogs and IL-31, we do see that, again, dogs that have allergic skin and ear disease are going to have more IL-31 circulating in their serum, as we are able to detect with current assays that we have available. We also see that dogs who have more IL-31 are itchier in general. And what's important is that dogs who have higher IL-31 are going to have worse CADESI scores. So their manifestation of their allergic disease, according to clinicians based on this standardized score, is going to be worse. So severity does seem to matter. If you have higher IL-31, you are probably going to have worse allergic disease. So knowing what we know, it's a soluble mediator called a cytokine. It plays a major role in the pathogenesis of canine atopic dermatitis. It's a peritogen, and it induces skin barrier changes. Do we think that IL-31 could be a therapeutic target for canine atopic dermatitis? Let's find out. Next lesson, that's what we're going to talk about.